Hello everyone. Before we dive into the details, let's first understand what exactly is sampling. Sampling is the process of selecting a small portion from a larger group. In this presentation, I will be talking about importance of sampling, the main factors that affect sampling quality, and also the basic steps involved in collecting a proper samples. Why is sampling important? Sampling is important because it's not practical to test every item in a large batch. It's just like taking just a few spoonfuls from a big pot of soup to check the flavor. In science and industry, it means collecting a part of material, product, or population to represent the whole. We do this because analyzing an entire batch is too expensive, time-consuming, or even impossible. If we do it correctly, we get results that reflect the entire batch. So next, we move on to factors affecting sampling. If the material is not homogeneous, meaning it's not evenly mixed, it's harder to take a sample that truly represents the whole batch. The sample size must be large enough to give reliable results. We also need to use the correct sampling tools and techniques to avoid contamination. In addition, the storage handling conditions must be suitable to keep the sample stable and in good condition until testing. Next, sampling follows a few important steps. First, we define the objective, which means knowing why the sample is being collected, such as for testing, quality control, or research. Second, we choose the right sampling method like random, stratified, or systematic sampling. Third, we select proper tools and containers that are clean and suitable for the material. Fourth, we collect the sample carefully, making sure it represents the whole batch. Finally, we label, store, and transport the sample correctly to keep it safe and ready to analysis.
Next, I will be explaining the basic sampling techniques that we use in analytical chemistry for solid, liquid, and gas sample, and also the precaution that must be taken for each sampling. I will be explaining about the first sampling techniques, which is solid sampling. The method that we use in this sampling techniques, the first one is coning and quartering, where the solid is grounded and mixed and then formed into a cone. The cone will be flattened and divided into a quarter to reduce the size of the sample evenly. The second method is grab sampling, which involves to take a small portion of sample from a different areas or spots from the solid bulks. Last method is drilling or coring. Is used for large or hard material like metal or soil. This method is useful to collect the sample from the inside of the material. The precaution that we must take for the sampling techniques is to use a clean or dry tool such as spatula or scoops. It is to prevent contamination or loss of fine particles from the sample. Next is liquid sampling that we mostly use for water, juice, milk, and oil. The method that we use in these sampling techniques, the first one is grab sampling, where a single sample is taken using a pipette, dipper, or bottle. This method works well for small containers and well-mixed liquid. The second is composite sampling. In this method, multiple small samples were taken over time or different depths, which is from top, middle, and bottom, and then the sample will mix together to form one representative sample. Last method is automatic sampling, which are used to collect sample continuously over a period. especially when monitoring changes in composition. The precaution that we must take were to avoid overfilling the containers with sample and also to use clean glass or plastic containers that were often sterile. The last basic sampling techniques is gas sampling. They are used in air quality, industrial process, and fermentation. The method that we use, the first one is gas-tight syringe. They are used to collect gas sample directly into an instrument. The second method is sampling bags, like Tetra bag. These are flexible plastic bags that were able to fill in with gas for temporary storage. They are used for environmental air sample and indoor air testing. The last method are evacuated canister. These are metal or steel containers with a vacuum that automatically open and pull in gas. This method is good for preserving the gas sample under pressure. The precaution we must take is to avoid contamination from air and to use proper materials such as glass and stainless steel that do not react with this gas sample.
Now, we'll talk about proper storage practices, which are very important in analytical chemistry. Proper sample storage is essential to preserve the integrity of a sample after collections. Inadequate storage can lead to contamination, degradation, or loss of important components, which affect the accuracy and reliability of analytical results. By storing samples under suitable conditions such as controlling temperatures, preventing exposure to air or light, and using correct labeling, the sample remains stable and suitable for analysis. First, to prevent contamination, we must use clean, dry containers. After preparing a sample, it should be sealed tightly to protect it from air, dust, or chemical fumes, which could be interfering with the analysis. Second, labeling is a must. Each sample must have a clear label that includes the sample name, date, concentrations if available, and storage instructions. This helps avoid confusion and using the wrong sample later on. Third, we must pay attention to temperature control. Some samples can be stored at room temperature, but others, especially light-sensitive or biological ones, should be kept in a refrigerator at 4 degrees Celsius to prevent decomposition of microbial growth. Next, we also need to protect samples from light and moistures. Light-sensitive samples like essential oil or iodine should be stored in amber-colored bottles. For samples that absorb waters from the air, called hygroscopic substance, we should keep them in sealed containers or in a desiccator. Also, we should always choose the right container material. For example, glass is suitable for acidic or organic samples, while plastic containers are safer for alkaline or corrosive substance. Lastly, it's important to record and organize all samples properly. Keeping a sample logbook helps us track the sample's age, purposes, and storage conditions. Organizing samples neatly by type or date also avoids confusions, especially during lab work. By following these practices, we can make sure our samples remain stable, uncontaminated, and ready for accurate analysis.
I will be explaining the advantages and disadvantages of sample preparation and dissolution, which is one of the basic requirements in chemical analysis. Let's start with the advantages. The first advantage is it improves accuracy and precision of analysis. Secondly, it helps remove interference substances. Thirdly, it allows analysis of solid or complex samples. Lastly, it allows the sample to fully dissolve for accurate titration or spectrometric analysis. However, there are also some disadvantages, such as it can take a lot of time, especially for hard samples. There is a chance the sample might get lost or contaminated. Sometimes, strong chemicals or heat are needed, which can be risky. If the sample doesn't dissolve properly, the result might not be correct. So, even though this step is very important, it needs to be done carefully to get good and reliable results. In conclusion, proper sampling ensures that the portion taken truly reflects the entire material, reducing bias and increasing representativeness. Sample treatment play a vital role in removing impurities or interfering substances that may affect the analysis. Correct storage conditions help prevent contamination or degradation, preserving the sample's original quality. Meanwhile, sample preparation ensures the material is in the right form and condition for accurate testing. Together, these steps enhance the accuracy, consistency, and overall reliability of analytical results.